I can see people in the chat. I don't see anyone on video. Is that what I'm supposed to see right now? Okay, great. So, okay. All right, so I have a short presentation to give on some of the basics about parasites, as well as a little bit about what I do and how I got into studying parasitology. So I will start that up, and once I'm done with that, I can come back over and then I'll be able to see the chat and answer questions. Does that sound okay? So let's see. I will start the presentation now. All right, can you guys all see my screen? Okay. All right, so I first wanted to start with just a brief overview of the definition of a parasite and then move on into some examples and some of what I find to be the more fascinating parasites. So a parasite is an animal that has a long-term interaction with another animal. So this is going to be something that occurs over days, weeks, or months. So something like a mosquito biting you wouldn't be a parasitic relationship, but something like a tapeworm living in your guts, that is going to be something that is a parasitic relationship. And in this relationship, the parasite is going to benefit while harming its host. And how much it harms the host can greatly range. So it can be something as little as it just causes some inflammation to actually causing extreme illness or potentially death of the host. However, most parasites aren't actually going to have that severe of a reaction, because if the host dies, often the parasite will die. So it's best for the parasite to keep the host alive. And there are two different parasites, types of parasites that we talk about. There are ectoparasites, which are those that are found on the outside of the body, and then endoparasites, which are found on the inside of the body. And I'll get into that with some more examples a little later on. Now, parasites have a very effective life strategy. And we know this because parasitism has evolved over 60 times in multicellular animals. And also, all the animals that you see on your screen now, all of them are host to many different types of parasites. And pretty much any animal you can think of is going to be host to at least one, if not many, parasites itself. Furthermore, even things like trees, they actually can be host to parasites. So they actually can have little root parasites and things like that. So why are parasites so effective? Well, it's because they can actually get another animal to perform some very basic life management things for them. So for like example, a tapeworm will live in the intestines of its host, and it doesn't have to worry about digestion at all because the host does the digestion for it. So it's getting other animals to do things for it, which actually saves the amount of energy that the parasite itself has to expend. So it's very, very effective. And now I want to show you a few examples of some parasites. And what I'm going to use as an example host is a lion. So lions, of course, are top of the food chain, carnivores, so they get exposed to a lot of different parasites. So some of those parasites are going to be on the outside of the body. Those are those ectoparasites I talked about. Those are things like ticks and mites and fleas. So this is something that you probably have had to deal with if you have dogs or cats. The other parasites that it gets are going to be those inside its body. So some of those parasites are going to be in internal organs, such as in the brain, the heart, the liver, or the intestines. And some of those parasites are things like tapeworms that'll be in the gut. There are things like hookworms that could also be in the gut. There are heartworm parasites that are a type of roundworm. There are flukes that'll occur in the liver, and then little thorny-headed worms that you can see here, which will be in the stomach. Then they can also have parasites in their blood. Often these are really tiny little parasites. Some of them are unicellular, so one cell, or they can be multicellular. So things like malaria. But there's also these little tiny blood flukes and things like what we call trypanosomes, and those cause some severe illness like sleeping sickness in South Africa. So often the things at the top of the food chain will have a lot of parasites. Another example of this is actually sharks, which is why I'm so interested in them. Sharks are fantastic hosts. They have over 1,900 different types of parasites that can infect them. Now, one shark will not have all of those parasites, but they can have many of them. And these parasites, as you can see here, can be found everywhere from the nose to the brain, the liver, the intestines, and even the skin. So they can have them pretty much anywhere. And they have, as you can see, a really great diversity of them. And so it gives you a little bit of an idea of how prevalent they are. And now that we've talked a little bit about the number, how many there are, and so on, why do we care, right? So parasites can actually be used for many different things. The first is they can help us understand if we have a healthy ecosystem, because a healthy ecosystem has many parasites in it. If we look at a system and find that there are parasites missing, that can tell us that something's actually wrong. So for example, some parasites can tell us whether there's pollution in the water. So sharks are known to have certain parasites in their gills. They typically have 3 different types of parasites in their gills. They have these basically little crustaceans here and little sea lice here, but they also have these guys here, which I'm going to zoom in on. That is a little flatworm, and it's got a really soft, squishy little body. So if it's in bad water, it's really badly affected. So if you look at a shark's gills and find this guy missing, that could tell you that there's something wrong, because whenever there's pollution, you won't find this guy. So it helps us to understand whether or not the water quality or the environment is in good health. Another reason that parasites are important is they can help us understand how the food web works. So some of these parasites, such as tapeworms or thorny-headed worms, are passed on through food. So if you know where these parasites are found as adults and as larval forms, then you can understand what animals are eating what type of prey. So for example, this is the common thresher shark, and we know that it has this little tapeworm in it. And this tapeworm is an adult in this shark. If we know which type of prey items have the larval form of this tapeworm, then we'll know what the shark eats. And in fact, we recently discovered that the larval form of this parasite is in this little scabbard fish. So that tells us that the thresher sharks are eating the scabbard fish. So it helps us better understand the ecology of some of these big predators. So now that we've talked about the diversity of parasites and the importance, I wanted to tell you a little bit about how I kind of got caught up in the crazy world of parasitology. So this is actually a picture of me from a conference this past summer. I go to a conference every summer. That's a meeting of all the American parasitologists, and I'm standing with a hookworm mascot. So I know he looks a little bit funny. And when I started out my career, I didn't know I wanted to study parasites. I actually thought parasites were kind of gross, and I didn't find them all that interesting in the beginning. So this was an unexpected path for me. I started going to my undergraduate studies at Florida Atlantic University, and I was studying actually marine biology. And I really actually wanted to work in sea turtle conservation at first. And I was working in a sea turtle hospital for a little bit, which was a lot of fun. But I decided after a while that I wanted to get some experience in the lab. I wanted to see if I liked research. So I applied for a few internships, and I was able to go to Oregon for a summer to do an internship with Dr. Kim Jacobson. And when I got this internship, I thought I was going to be studying the interactions of native and invasive fish in the Oregon River. And I was going to be working with these fish, but when I got there, I found out I was actually going to be working with the parasites of these fish. So basically what I had to do for this project is I had a bunch of specimens of each of these types of fish, and I looked at the parasites in them. So I dissected them, looked at all of their organs and found as many parasites as I could. And I ID'd all of those parasites. And what I was trying to do was understand whether the native fish, so those that belong naturally in that region, had more parasites than the invasive, so those fish that was introduced to the region. And I wanted to understand that because there's a theory that the reason that these new invasive fish are able to outcompete the native ones is because that they have fewer parasites. So they have less of a drain on their resources. So I went through, I did the dissections, I collected, identified my parasites, and I found that there were 6 different groups of parasites in these native fish, but only 3 in the invasive. So that actually fit with the theory. It does seem that the invasive fish has fewer parasites, and therefore that might be part of the reason that they're able to compete better than the native fish. So after this project, I was kind of hooked. I really liked parasites, and I decided to go get my PhD at the University of Connecticut working with shark tapeworms. And I decided on sharks just because, as I showed you before, sharks are really great hosts, and I think tapeworms are one of the most interesting parasites out there. So let's talk about what a tapeworm is really quick. A tapeworm is a type of flatworm, and it has 2 regions to its body. This upper region here, that's kind of like the head region, that is called a scolex, and basically that's where it attaches to the intestines of the sharks. And there's many different ways that it can attach. So it can have a sucker, kind of this opening you see up here. They can have a sticky pad on their head. Some of them have these muscular grabbing apparatus, so basically little pads that grab onto the sides of the intestine. Some have these really nasty hooks that you see here, and some have tentacles with hooks all over them. So there's a lot of diversity in attachment structures in the tapeworms. The rest of the body, you see here, are basically different little packages that contain eggs, because these guys have to produce a lot of eggs over their lifetime. And that is because tapeworms are passed on through their hosts through their food. So this is an example of a tapeworm that infects big carnivores. So this tapeworm is an adult in this bear, basically. When it's in the intestines of the bear, it produces a lot of eggs, and the eggs are basically pooped out by the bear. Once the eggs are in the water, it opens up and releases this first larval form that's then eaten by this tiny little crustacean. When the crustacean eats it, it goes into its muscle and kind of just hangs out until this little crustacean is eaten by a fish. Once in the fish, the larval form moves into the muscle again, becomes the second larval form, and then again hangs out until it's eaten by a big predator, or us, which is why it's very important to cook your food. If you cook your food, you won't get parasites. And it'll become the adult in the intestines here. So what I work on are the tapeworms of this shark here, the pelagic thresher shark. It can get to be about over 200 pounds and over 10 feet long. And it actually uses this really long, funny-looking tail to stun its prey. And we've known for a while that it has 3 different types of tapeworms in its intestines. And we know all of these tapeworms are very closely related because they have suckers, and they all have segments on their head region. Now, recently we found another type of tapeworm in the intestines of this shark, but it's really strange looking because it doesn't have suckers or those segments on its head region. It doesn't have any characteristics at all that are similar to these guys. However, when I looked at the DNA of this one, this new one, and compared it to the DNA of all these others, I found that they're actually closely related. And in fact, this tapeworm and this tapeworm are sister to each other. So the question that I'm trying to answer with my research is, how did this worm evolve to have such a different body than its close relatives? And in order to go about answering that, I actually traveled to Taiwan a few years ago to collect more tapeworm specimens. And in Taiwan, basically you get the sharks and the fish from these big fish markets where you can go around and basically buy the fish from the fishermen in the market. And then we went and we would take the shark or the fish back to the lab. We would open it up, pull out the intestines, and then we would take the tapeworms out of the intestine. And this is actually an example of that weird new tapeworm alive in a dish from one of those intestines. So you can see it moving around there. And this guy is pretty unhappy. It would really rather be in the intestines of a shark than in this little dish. So now that I had all the tapeworms that I needed, let's see, there we go. I could start to answer my questions. And the first thing I needed to do is I wanted to better understand how this tapeworm is different from its sister. And I looked at it using this really big, powerful electron microscope to look at the cells on the inside of this tapeworm to better understand how it differed in its body form. And then I also extracted all of its DNA so that I could compare the DNA of this one to all of its sisters in order to see what could have changed to allow it to have this different body form. So that's all I have for a presentation right now. So I'm happy to answer any questions that you guys have at this time. Is anyone still there? I'm not seeing anything come up on chat. Okay. What are some of the other parasites I've studied? So some of the other parasites I've studied are, I've looked at a blood parasite that infects oysters and mussels. And it's very problematic because it actually can kill all of these oysters and mussels, which are a big fishery down in Florida, which is where I did a lot of my studies. So I've studied them, and I looked at what different types of oysters and mussels that they're in. And I also looked at whether they had any seasonality to them. So some occur more in the summer? Do they occur more in the winter? And I found that the parasites tend to occur more in the summer and the spring and less in the winter. Let's see, another question is there a difference between freshwater and saltwater parasites? So there are ones that, so there's like things like tapeworms can occur in both saltwater and freshwater, but different types will be found in the different environments. So usually we can tell where an animal has been based on the type of parasites it has. And part of the way that they actually discovered that both sharks can go in fresh and salt water is by discovering that it had freshwater parasites in it. That's what kind of led them to first investigate whether it was actually just eating somehow some freshwater fish or actually traveling in freshwater itself. Let's see, the next question is, can a parasite be host to another parasite? Yes, actually. It's kind of rare, but it's called hyperparasitism. And we've actually seen it a few times in the lab where we'll find basically juvenile tapeworms attached to some of our tapeworms. And there can be some parasites within things like the external parasites, such as fleas and ticks and mites and things like that. So it is possible as well. So tapeworm, how do tapeworm genomics compare to human genomics? The tapeworm genome, so basically all the DNA that animal has, is actually much smaller than the human genome. It is about 1/4 of the size. And we think that might be partially because tapeworms have a tendency to get rid of things that they don't need. So actually tapeworms don't have things like sensory systems or a digestive tract. So it's actually been able to get rid of all the genes that have to do with those, so they have a smaller genome, which is kind of cool to look at. Are you still interested in being a marine biologist? I actually still do consider myself a marine biologist. I'm just kind of working in a very small field within marine biology, since I am still working on the evolution of parasites of sharks. I am trying to stretch myself out a little bit to include some freshwater systems, though, as well. So I do consider myself a marine biologist. It's just a very very different view than I think most people think of a marine biologist, because there's so many different things you can do with marine biology. It doesn't always just have to be fish itself or things like that. It's a very expansive field. How do you get rid of parasites if you have one? For most things like tapeworms, you can take one pill and it basically dissolves it. So tapeworms are actually really easy to get rid of. With other things, sometimes you need to do surgery depending on the parasite, but many of them, it can just be like a little pill that you take, and that'll take care of it. What happens to the tapeworms after you are done with your research on it? So for my studies, I have to basically grind up the tapeworms that I used to get their DNA. So basically there's nothing left of them when I'm done with them. I do have some like extra tapeworms that I'll keep with me just in case I need to do further studies in the future. But those basically I have preserved in ethanol that I keep in case anything else needs to be done in the future. But most I kind of grind up or grind up or cut up if I'm doing the microscopy, looking at them with a microscope. So there's not much left of my tapeworms, unfortunately, once I'm done with my studies. Any other questions? Let's see. Are shark tapeworms similar to dog-cat tapeworms? So they are somewhat similar, yes. So, but they're in two different groups altogether. So they're like two different families, basically. And the ones that are in dogs and cats tend to have a much more simplified attachment region. So usually they just kind of have a few hooks or some suckers, versus the tapeworms in sharks tend to have a lot more different like spiny attachment or sticky attachments. However, I will say the ones in dogs and cats tend to get bigger than those in sharks. Overall, how big can tapeworms get? So the ones that I work with are pretty small. The biggest one I work with is about that big, so only a few inches. However, tapeworms can be several feet long, especially the tapeworms from things like dolphins and whales. We have a collection facility at UConn where we have a jar that's about this big that is pretty much just all one tapeworm. So they can be many, many feet long. So what other marine organisms might the thresher shark tapeworms be found in? So that's actually really interesting. The tapeworms in the thresher shark are only found in the thresher shark. So tapeworms of sharks tend to be very specific for their final host. They can be found when they're in their larval stages, they can be found in many different types of fish or crustaceans, but when they're adults, they're only found in one type of shark, actually. So that's part of how we can actually identify sharks sometimes if we're not sure what we're working on, is if we look at the tapeworms, because we know what type of tapeworms can be in which type of sharks. How often do parasites evolve? And did you ever find your answer about the evolution, about the tapeworm you spoke about? So we think that it's likely that they're kind of evolving continuously, but it takes many generations to see that. So we think that they're still evolving fairly recently. The new species, the new tapeworm that I talked to you about, the weird looking one, we think that happened actually very recently, because that's what our DNA tells us, is we can kind of gauge how many the number of differences in the DNA can allow us to gauge how much time has passed. And the amount of changes in the DNA is showing us that this happened fairly recently. So we think that's continuing to happen. How often, and the second part of your question, so I'm finding that for the tapeworms I'm looking at, it looks like this new type of tapeworm evolved within this shark, and we're seeing some differences in their genomes in that this weird species, it looks like there's many different places where the DNA has been moved and then flipped. So we think that might be part of what's causing it. We're also seeing some genes are being used, so expressed, basically turned on more often in this weird species than it is in the normal species, or the normal tapeworms. What are some specific parasites that can be bioindicators? So things like the thorny-headed worms, they're these little worms that occur in the digestive tract that have little spines on their head. They're actually great bioindicators for metal accumulation in the environment. So because we can use them if we test the metal levels, the heavy metal levels in those parasites, it tends to tell us how much heavy metal is present in the environment and in the water. So those have been used to test whether there's heavy metal contamination within an environment, which is pretty neat. Other ones that have been used as bioindicators are things like ticks and mites have been used to understand how and where animals have been traveling. So if we know the range of a certain type of tick, we know that if a host, such as a bear, has that kind of tick, we know it went through that region. So it can tell us where these animals are traveling. Is there evidence that parasites were ever able to exist without a host? If so, how did they live? So many of these parasites, we think, evolved from free-living animals at one point. So for example, tapeworms, the tapeworms come from a family, basically, where there's 3 different types of parasites within that family, but they also have a free-living relative. So we think all those parasites likely evolved from that free-living relative, but that probably happened many of hundreds of millions of years ago, probably around 300 to 400 million years ago, likely. And they probably lived in we think, especially for things like the tapeworms, they lived probably in shallow water, I think marine environments, most likely, is our guess for the tapeworms. But everything that we've looked at are somehow related to free-living species, so we think all the parasites at one point came from free-living species. And it depends on what we're looking at, how they lived. Any other questions I can answer? These are all great questions, by the way. Anything that I missed? Okay, here we go. Does that turning and flipping of DNA expression have anything to do with DNA methylation and possible environmental stressors that were placed on tapeworms' ancestors? So I'm not sure about the DNA methylation. I haven't gotten to that point of my analyses that I'm able to tell that yet. I can only look at some of the bigger pictures yet. I'm hoping to get to that a little bit later in my research. And we don't think that there might be environmental stressors that cause this, because all of these tapeworms are occurring in the same environment because they're all within the same shark. So any environmental pressure that one is feeling, all of them should feel. So we don't think it's anything to do with the environment per se. We think it's more likely it was something where there was a mutation that occurred that allowed for this new attachment strategy to evolve, basically, and it was probably a mistake at first, but actually the attachment strategy used by this new weird tapeworm actually is a much better attachment strategy than is used by its closest relative and its sister. So it was most likely something like that, but we don't think it was the environment, but there's many more tests that I kind of need to run before I can make a very firm conclusion on that. Yeah. Thank you so much for having me. This has been really fun. I hope I was able to answer all your questions reasonably well. And they were fantastic questions. Is there anything else that I can talk to you guys about, or? You all have a great afternoon as well, and happy Parasite Day. It was nice talking to you all.